Hey everyone, welcome to my YouTube channel. So in this video we will discuss about the histochemical classification and identification of total proteins and glycoproteins. So in this we will identify the proteins by using Bromophenol blue and Congo red method. So first of all we have Bromophenol blue method. So first of all, introduction. If we desire to identify the proteins, so we have two basic techniques. The first is specific characteristics like molecular weight, solubility, chemical bond, etc. And second method, which is the mostly used to visualize the proteins in histological section. The second is the specific amino acid present. So we use the second method as comparison to specific characteristics. Okay, next we have two factors. So there are also two factors. First is the presence of functional group and second is the formation of functional group on this side chain of amino acid. So these functional group react with the stain, or we can say the reagent, and give the colored result. We can see in the histological stain in Bromophenol blue and Congo red method, we will see the end result. So in Bromophenol blue, the end result is blue. Okay, so therefore it is a mercury Bromophenol blue. Okay, so let's start with the histological stain of proteins. First we will do the principle, then we will see the preparation, and last we will see the staining procedures and result. So the principle of Bromophenol blue is, it is a anionic dye, phenol phthalate anionic dye. We can see the structure of mercury Bromophenol blue. So this Bromophenol blue bind with the protein under the neutral conditions or acidic condition. So in this way we can determine the proteins in sample, and we can say especially the cell membrane proteins. Okay, mercury Bromophenol is used for the detection of total proteins including glycoproteins. And if we talk about the advantage, so we can identify even low amount, very little amount of protein in a sample by using the mercury Bromophenol blue stain. And basic proteins like histones, transcription factors, and ribosomal proteins, we can use these proteins, which are the basic proteins, which bind to Bromophenol blue dye even in the absence of mercury. Okay, if we use only Bromophenol blue, not mercury Bromophenol blue, we use here Bromophenol blue with the basic proteins, so basic proteins can easily bind with the Bromophenol blue. Okay, the third step is the non-basic protein. Firstly we talk about the basic protein, now we are talking about the non-basic protein, also bind with the Bromophenol blue dye but by coupling through mercury. So basic proteins can bind in the absence of mercury, and non-basic proteins only bind with the presence of mercury. Okay, and the amount of dye depends upon the proteins. Okay, the dye bound is proportional to the amount of protein. How much protein we use, then it will proportional to the dye. Okay, if we use 1 gram protein, that means 1 gram dye. Okay, so next we have basic proteins. Why we use basic proteins? Mostly because basic protein have strong tendency to bind the Bromophenol blue, because Bromophenol blue, we all know that it is an anionic dye. We already discussed anionic dye means it carries the negative charge, which shows the electrostatic attraction to the positive charge. Positive charge contains the basic protein, so positive, negative, there is a electrostatic attraction, therefore basic protein can bind to Bromophenol blue dye even in the absence of mercury. Okay, this is the structure. Next we will see the preparation of mercury Bromophenol blue. So there are two methods. First we have dissolve the Bromophenol blue, approximate 10 milligram in 100 milliliter of 10% mercury chloride in 95% alcohol. Second we dissolve the 1 gram mercury chloride and 0.05 gram Bromophenol blue in 100 milliliter of 2% of acetic acid. Okay, next we have staining procedure. We have 4 steps. First steps we We will do the deparaffinize the tissue sections, which are dehydrated in absolute alcohol, and then we will do incubate for 15 minutes in mercury bromophenol blue at room temperature. Second, we will rinse with 0.5% acetic acid, then remove the extra stain for 5 minutes, and wash with tap water until the color become blue. Then in the third step, the sections will dried, and it will clear with the butanol and xylol. Then we will mount with DPX. We all know that DPX as a mounting medium. Then last, it if there is a presence of blue color, that means the proteins is present. Okay, this color. So this is the liver section stain with mercury bromophenol blue.